So let's now talk about sex-linked characteristics, which represent one of the major extensions of Mendelian principles of inheritance. Sex-linked characteristics are determined by genes located on sex chromosomes. Therefore, genes which are located on X chromosome determine X-linked characteristics, and those located on Y chromosomes determine Y-linked characteristics. You recall in previous recording we discussed differences between males and female sex chromosomes, and these differences in sex chromosomes also determine different pattern of inheritance of sex-linked characteristics. First person who noted and explains these differences for sex-linked inheritance was Thomas Hunt Morgan. He was an embryologist. However, after the discovery of Mendel's principles of inheritance, he became inspired and extremely excited about genetics. And he started to perform genetic crosses first with rats and mice, and later on he switched to fruit flies. When he moved to Columbia University, he was assigned a small laboratory which he turned to so-called fly room. You can see it right here. It was a room which was avoided by majority of staff at Columbia, and there were many funny stories going on around this room. It was a room which was crowded not only with his students but also with fruit flies which escaped, and which was extremely smelly from food they prepared for fruit flies. Nevertheless, in this room, Thomas Hunt Morgan and his student made important discoveries which explained behavior and inheritance of sex-linked traits. Studying fruit flies, Thomas Hunt Morgan and his student discovered a rare mutation which caused white eyes in fruit flies. If you know fruit flies, if you study fruit flies, you know that wild type phenotype is red eyes, and it's very easily recognized feature. They observed first few males who carried this mutation. However, later on they also observed few females carrying mutated white-eyed phenotype. So they become extremely interested in this mutation and the mechanisms by which it might be inherited. So they designed a crosses in which they first crossed red-eyed wild-type females with mutated male who had white-eye phenotype. F1 generation of these crosses yielded uniform results. All offsprings were red-eyed. They all expressed wild-type phenotype. This was not surprising considering Mendel's law of inheritance. So if the red eyes are dominant over white eyes and you cross two purebred individuals, you expect that F1 generation will be uniform. However, surprise came when they performed reciprocal crosses in which they crossed white-eyed female with red-eyed males. Under this condition, F1 generation yielded surprising results because all females in F1 generation were red-eyed, so they were wild type, while all males carried mutation or expressed mutated phenotypes. These were all white eyes. Now, if you compare results of these reciprocal crosses, you see striking difference. Now, keep in mind that if you follow trait, which is inherited by genes or which is encoded by genes located on autosomes, reciprocal crosses yields the same results. So under such condition, it does not matter whether it is mother or father, which parental individual, whether it is maternal or paternal individual who carries mutation. However, under this condition, it did matter which parent carried mutation. So to define these unexpected results, Thomas Hunt Morgan and his student went further and performed crosses in which they crossed offsprings of F1 generations. You recall, this site has original parental phenotype and genotypes is outlined here, and this is original parental genotype outlined for this cross. And again, they obtained very surprising results. If they crossed F1 individuals from F5 generation, which were descendant of cross where female was originally wild type and male was mutated, F2 generations of this F1 offsprings, they cross these offsprings, F2 generation yielded surprising results because all females were wild type phenotypes and half of males carried mutated phenotypes. This was not expected. What they expected was that there would be equal proportion of wild type and mutation in male and female. Reciprocal crosses were even more striking. If original parent in original cross where in parental generation female was mutated, and you recall in F1 generation there was all female were wild type and all males carried mutated phenotypes or showed mutated phenotypes. When they cross this offspring from F1 generation, they obtain ratio of red-eyed individual to white-eyed in ratio 1:1. And within those, if you break this F2 generation into male and females, you see that half of males and half of females carried red eyes and half from each sex group carried white eyes. So this results of F1 and F2 generation clearly demonstrates a difference in inheritance of white-eyed mutation from regular autosomally encoded traits. So based on this data, Morgan proposed that the locus for eye color is on X chromosomes. So therefore, female has 2 X chromosomes, so it can be either homozygous dominant, homozygous recessive, or heterozygous. So heterozygous would manifest a wild-type phenotype. However, male only carry 1 X chromosome, so it is hemizygous. So even 1 mutated allele will be manifested. So let's now examine X-linked characteristics in humans. There are 2 disorders which are very well studied in humans, which are X-linked: red-green color blindness and hemophilia. So let's focus on red-green color blindness. You know that we perceive the light through the light-sensing cone which are located in eye retina. Each cone contains one of the 3 pigments. One of these pigments can capture blue light, and gene for this pigment is on autosome. Another cones can capture green light, and third one can capture red light. So the gene for pigments that capture red and green lights are located on X chromosomes, and mutation in these genes causes red-green color blindness. So let's follow these crosses. Let's follow the scenario. First, let's assume that a woman who has normal vision and there is no history of color blindness in her family, which would indicate that she is homozygous dominant, so she has wild type alleles for red-green pigment, marries a man who is colorblind, so he has his X chromosome carries mutated allele. So what is the probability that these two parents will have children who will be colorblind or who will be carrier? So what kind of gametes female makes? She only makes gametes which carries X chromosome carrying wild type allele. Male can form two types of gamete, either gametes which carry X chromosome with mutated allele or gametes which carry Y chromosome. So, what are the probability, what kind of genotype and phenotypes their children could have? So, all girls, if you see, all girls will have normal vision, however, they will be all carriers. And all boys will have normal vision. So you see, under this scenario, all children of these parents will have normal vision. However, all girls will be carriers. So in F2 generation, there is 50% chance that they will pass recessive mutated allele located on this X chromosome to their sons. And in case that son carries this mutation, they will be colorblind. You can see it in reciprocal cross. Let's consider that under this scenario, female is colorblind and she marries a man with normal vision. So again, female can form only one kind of gametes which carry X chromosome with mutated allele, while male forms two kind of gametes, one which carries X chromosome and one which carries Y chromosome. So what would happen, what is the probability of their children being colorblind? Again, if you look for girls, all girls will have normal vision. However, all of them will be carriers, while all boys will be colorblind. So why it is? Keep in mind that males are hemizygous. So in order for female to manifest X-linked characteristic, she has to be homozygous recessive if the characteristic is encoded by recessive allele. However, in male who is hemizygous, he possesses only one X chromosome and there is no homologue carrying homologous allele. Inheriting one mutating allele is sufficient in order for him to manifest mutated phenotype. That's why X-linked characteristics such as red-green color blindness and hemophilia are more prevalent among males than among females. Let's now look at Z-linked characteristics. You remember that organisms with ZZ-ZW sex determinations have inverse determination of sex as XXXY. In this system, there are females who are heterogametic and males are homogametic. So female inherits W chromosome, which W chromosome has similar function as Y chromosome in humans. So how does this reflect into the inheritance of Z-linked characteristics? The inheritance is the same as that of X-linked characteristic, except, keep in mind, except that the pattern of inheritance in males and female is reversed. So what applied to inheritance of X-linked traits for males. Now under Z-link characteristic it would apply for females. Now here is one of the example which follows the color of the feather color in Indian blue peafowl. Blue, which is dominant, over cameo, which is recessive. So solving this problem will actually clarify for you how this reverse pattern of inheritance apply to Z-linked characteristics. So we discussed X-link and Z-link characteristics. So let's now focus a little bit on Y-link characteristics. Y-link characteristics are encoded by genes which are located on Y chromosome. So their inheritance is very simple. Since Y chromosome is always passed from father to son, those characteristics will always be passed from father to son. And actually they are used as a Y-link genetic markers when you follow the traits which are passed from father to son. Now, also, the inheritance of Y-link characteristic is very simple. The history and evolution of Y and X chromosome is very interesting, particularly Y chromosomes. We know that both X and Y chromosomes were evolved from autosome. Somewhere around 250 million years ago, one of the autosome acquired new gene, SRY gene, which we know that is gene determining maleness in humans nowadays. So afterwards, throughout the evolution, this pro-Y chromosome accumulated additional mutation, which actually acquired more features to males that enables them to reproduce, to be more competitive and have increased fitness. So most genes, Y-link genes, play a role in male sexual development, fertility, mating advantage, and so on. However, last year, towards the end of last year, there was a paper published online, which will be published this year in Nature Genetics, which actually stressed that situation is not so simple, that loss of Y chromosome in males in certain cells of male, loss of Y chromosome increased chance of cancer. So obviously we underestimated the function of the genes located on Y chromosomes. Why it happened, why it is, because if you analyze Y chromosome, you see that 2/3 of Y chromosome is formed by heterochromatin. Heterochromatin means that there are no active gene, is constantly condensed. Now, if you look on genes which are encoded on Y chromosome, you find only approximately somewhere around over 300 genes which are active on Y chromosome, which is very low number in comparison to 1,000 or more genes located on other chromosomes. So, Y chromosome has large sequences which are not active. Another feature which is very surprising, which is linked to Y chromosome, is presence of 8 massive palindromic sequences. What are palindromic sequences? You remember in English, palindrome means that you can read word from front and from back, such as rotor. In genetic, palindromic sequences mean that there are homologous sequences on chromosome. Let's say, try some theoretical sequence. When we start 5', let's say ATGC sequence, and you recall we have anti-parallel strands. So on second strand complementary sequence would be TACG. So where is a palindrome? You have to define palindrome due to the anti-parallel nature. You can read palindrome only on the complementary strand. So it will be CGTA. Why? Look, here is a 5', to 3' sequence is ATGC. So here it is, you can read 5' to 3' only complementary on complementary strand. So it would be GCAT, and the same apply here. If you look for 5' sequence, it's GCAT, and here GCAT. So these palindromic sequences, why they are important? Remember that during meiosis 1, homologous chromosomes undergo recombination due to the crossing overs. Now, that apply also to X chromosomes, two X chromosomes. However, X and Y chromosomes, they have only very short pseudo-autosomal region which are homologous. So there is very low frequency of recombination between them. Now, if Y chromosome does not undergo recombination, what does it mean? It would accumulate many mutations. Some of them can be deteriorating. So this feature or this reason was actually found behind the shortening of Y chromosome over the period of time. Now, how palindromic sequences can help? Y chromosome actually can recombine. It can, his sister chromatid can recombine due to the palindromic sequences, as well as there were some papers published which show that a Y chromosome can form secondary structures, loops, where palindrome enable recombinations. So this recombination plays important role in avoiding accumulation of mutations. However, every benefits might come with some opposite results, and it might happen also that during cell recombination of Y chromosome, certain genes might be lost. For example, a SRY gene might be deleted. However, when I mentioned that Y chromosome is shortening, and it was actually proposed that in several million years Y chromosome will disappear. So individuals with Y chromosome be happy. Recent publication actually claims that this degeneration and shortening is slowed down, so there is no reason to consider disappearance of Y chromosome. At least one news here. The last thing I would like to discuss is dosage compensation, which equalizes the amount of protein produced by X-linked genes in males and females. Why do we need dosage compensation? You recall that genes which are located on X chromosome encode production of proteins the same way as the production of protein is encoded by genes located on autosome. However, we encounter a problem because in males there is only one X chromosome, while females have two X chromosomes. So the produced dose of protein encoded by this gene in female would be double. Since proteins are involved in metabolic pathways, the amount of the proteins have to be balanced, and this is particularly important during embryonic development. So evolution come up with solution through dosage compensation, which might be achieved differently. In some organisms, this compensation is achieved by doubling the activity of gene located on X chromosome in males. In another organism, it might be decreased activity of genes located on X chromosome in females. In humans, dosage compensation is achieved through inactivation of one X chromosome in female who has two X chromosomes. This inactivated chromosome appears in the cells of female as a dark stained body, which is called bar body. And this was observed very early in 1949. However, only in 1960-61, Mary Lyon proposes this dark bar body represents inactivated X chromosome, which we call bar body, and it is referred to as a Lyon hypothesis. So, what do we have to know about this inactivation? Few facts. Firstly, we know that X inactivation take place very early during embryogenesis. And the reason is because, as I mentioned before, the balanced production of proteins during embryonic development is critical. What we also know is that this inactivation is completely random. What does it mean? Let's say the female is heterozygous for the, let's say, gene A. Now, we all start as a one-cell zygote, so during embryogenesis there are divisions after divisions of cells, and very early during this division, in one of these descendant cells, one of these X chromosome will be randomly inactivated. Let's say in these cells, chromosome carrying dominant allele will be inactivated, and chromosome which carries recessive allele will stay active, while in another cells, randomly opposite chromosome will be inactivated. So here you will have chromosome which carries dominant allele which stays active, and that one which carries recessive is inactivated. So as division and replication of cells and differentiation goes on, every single cell which is descendant of this particular cell would carry the same allele. Okay, so this will form patches which would carry only this recessive allele on X chromosome, while cell descending from this cell all will carry dominant allele. Okay, so now there is one problem which you should have noticed. We talk here that X chromosome, one of the X chromosome in female is inactivated and form heterochromatin, from which the genes are not expressed. However, just before we were discussing X-linked characteristics such as red-green color blindness and hemophilia, which are more prevalent in male because they are hemizygous, while female can be heterozygous. So how does this all goes along? By there is very simple explanation, which was proven that X chromosome, which is inactivated, is not inactivated completely. Only approximately 75% of the chromosome and corresponding X-linked genes are inactivated in every female. There is about 15% of genes, 15% of X chromosome stays active. So genes located on this portion of X chromosome are expressed on both X chromosomes. Remaining 10% varies. Remaining 10% might be active in some female and inactive in another females. So basically this triggers pattern which we will define later in next slide. One of the best studied random X inactivation is patch distribution of coat color in tortoiseshell and calico cats. Those of you who love cats, you know that color coding in cats is encoded by complex mechanisms, so it's kind of complicated and there are many genes involved. So we will simplify it and we will just focus on X-linked genes which encode black color and genes which encode orange color. So you know that males, since males are hemizygous, can be either black or orange, but under normal circumstances you don't find patchy males. However, females who are heterozygous and during embryonic development random X inactivation take place might have patches of fur color which are black and patches which are orange, and it's because during embryogenesis in certain cells the X chromosome carrying allele for black color was inactivated, while in another cells X chromosome which carry allele for orange color was randomly inactivated. Now We know about random X inactivation. We know that it happens. We have examples, many others. Now, question is, how does it happen? What are the mechanisms which assure random X inactivation? So, first thing which we have to consider is how cell counts number of X chromosomes. How cell can assess how many X chromosome does it carry? And it does have this mechanism. It must have this mechanism because if you look at normal female, in normal female, only one X chromosome is inactivated. In polyfemale, which carries three X chromosomes, two of them are inactivated. In male, which is normal XY, there is no X chromosome inactivation. However, in Klinefelter syndrome, there is inactivation of one X chromosome. If male is Klinefelter, very rare, which carries three X chromosomes, two of them will be randomly inactivated. So question is, how cell assess number of X chromosomes? And the answer is we don't know. Really, we don't know how it happens, but it does happen. Second question is, how cells select which chromosome will be inactivated? And the answer is there is no selection. It's completely random. In one cell, one chromosome can be inactivated randomly, in another chromosomes. Third question which we have to pose is how it happens. And here there is involvement of the gene which is called ZISE gene, X inactivation specific transcript, respectively gene which encode X inactivation specific transcript. So, both X chromosomes carry gene for ZISE. So ZISE gene is present on both X chromosomes. During early embryogenesis, this gene is become activated on one of these chromosomes. Let's say it's activated on this chromosome. So it encodes synthesis of long non-coding RNA. So the product of ZISE gene is RNA, which interacts, since it's homologous, it actually hybridizes with X chromosome from which it was transcribed, and it attracts metal transferases which alter epigenetically histones associated with this X chromosome. We will talk about this mechanism in more details when we will talk about epigenetics. So, this epigenetic change in activation of one of the X chromosomes, you recall 75% of this chromosome, will be passed to every somatic daughter cells which descend from this chromosome.